Today, I want to share some valuable insights into GMP cleaning and sanitation practices in the pharmaceutical industry, an essential topic for ensuring product quality, safety, and compliance. Whether you're a quality assurance professional, a production operator, or just interested in how pharmaceuticals maintain their high standards, understanding these practices is crucial. First, let's talk about what GMP, good manufacturing practice, entails in cleaning and sanitation. GMP guidelines are strict protocols established to prevent contamination, cross-contamination, and ensure that medicines are consistently produced to quality standards. Cleaning and sanitation are foundational aspects because they directly impact product integrity. So what does GMP cleaning mean? GMP cleaning involves removing dirt and residues through mechanical or chemical means. After cleaning, inspect visually, then test samples in the lab to ensure cleanliness. Use appropriate detergents and conditions. A key concern is ensuring cleaning agents don't damage equipment or compromise product quality. Proper GMP cleaning maintains safety and effectiveness in pharmaceutical manufacturing. What is sanitation? Sanitation reduces microbes using chemicals, heat, steam, or scrubbing. Proper cleaning is essential first, then, sanitizers lower microbial levels. Effectiveness depends on microbes, material, concentration, temperature, pH, and duration. Always remove sanitizer residues after use and follow guidelines for best results. Clean first, sanitize second for safety. Contaminants in pharmaceuticals. Contaminants, chemical, environmental, and biological, threaten drug safety and quality. Chemical contaminants include impurities, heavy metals, and residual solvents. Environmental ones involve microbes, dust, and pollutants. Biological contamination covers bacteria, fungi, and endotoxins. Mitigation includes strict testing, GMP, sterilization, and environmental controls to ensure safe, effective medicines. Chemical contamination. Chemical contamination occurs when the product becomes contaminated with other foreign materials, products, or residues. This can occur when different products are processed in the same equipment or when materials such as cleaning agents and lubricants come into contact with products or equipment surfaces. The way to avoid chemical or biological spills or contamination is by strictly following written GMP cleaning procedures. Often, you have to validate the steps and conditions in the procedures to ensure they are effective every time under different conditions. Changing the steps or conditions may make the cleaning process ineffective. Some areas in the facility pose higher contamination risks, especially where products are exposed, like dispensaries, formulation, and filling rooms. To prevent contamination, use filters to control air quality and perform regular cleaning and monitoring. Other zones, such as storage or secondary packaging, have lower risk since products aren't directly exposed. These areas require less frequent cleaning and monitoring. Stay vigilant to maintain product safety. Biological contamination. Biological contamination refers to contamination by bacteria, yeasts, molds, viruses, or any other microorganisms that may be present in the product. This also can include residues from dead microorganisms or endotoxins, which can contaminate injectable products. The origins of biological contamination vary but include the environment, personnel, raw materials, contaminated water, and poorly cleaned or wet equipment. While cleaning reduces biological contamination, sanitizing agents are important in killing microorganisms. When you are in business to prepare sterile products, production must be conducted in cleanrooms protected by high-efficiency particulate air (HEPA) air filters. Cleanroom requirements are very strict in these areas and are usually associated with positive room pressures, overpressure, to protect the areas from external airborne contamination. Usually, 99.97% of all airborne particles above 0.5 microns are excluded by the HEPA filters. Cleaning agents can be broadly categorized based on their chemical nature and specific functions. Alkaline cleaners primarily contain substances like sodium hydroxide, NaOH, and potassium hydroxide, CO, which are effective in removing grease, fats, and proteinaceous residues. Acidic cleaners include agents such as citric acid, phosphoric acid, and nitric acid, and are typically used to eliminate mineral deposits, scale, and inorganic residues. Detergents are classified into non-ionic and anionic types. Non-ionic detergents are gentle and effective in removing oily soils, while anionic detergents contain negatively charged ions that enhance cleaning power against dirt and grime. Solvents like isopropyl alcohol, ethanol, acetone, and ethyl acetate are utilized for dissolving oils, adhesives, and certain residues. Chelating agents, notably ethylene diamine tetraacetic acid, EDTA, bind metal ions to prevent or remove mineral deposits. Enzymatic cleaners incorporate enzymes such as proteases, amylases, and lipases to break down proteins, starches, and fats, making them especially useful for cleaning biological residues. Surfactants like Tween, Triton X100, and sodium lauryl sulfate reduce surface tension, helping to lift and remove soils from surfaces. Ammonium compounds, exemplified by benzalkonium chloride, serve as disinfectants with antimicrobial properties. Phosphoric acid-based cleaners are specialized agents that utilize phosphoric acid for targeted cleaning applications. Water for injection, WFI, or purified water is used as a solvent or rinsing agent to ensure cleanliness without introducing contaminants. Finally, aldehyde-based cleaners, including glutaraldehyde and formaldehyde-based solutions, are employed for sterilization and disinfection purposes, especially against bacteria, fungi, and spores. These diverse cleaning agents are selected based on the nature of residues or deposits to be removed and the specific cleaning requirements of the application. Sanitizing agents. Different sanitizing agents will be effective only in certain circumstances. We have categorized 4 types of sanitizing agents commonly used in pharmaceutical manufacturing. 1. Sanitizers. Proper use of sanitizers can reduce bacteria by greater than 99.9% if used on pre-cleaned surfaces. Pre-cleaning is always required to reduce bioload and dirt. 2. Disinfectant. Proper use of disinfectants can result in 100% killing of vegetative bacteria, target viruses, and target fungi. Pre-cleaning may or may not be required, but it is considered a prerequisite. 3. Sterilant. Proper use of sterilants can result in 100% killing of all microorganisms, including bacterial spores. Pre-cleaning is always required to reduce bioload and dirt. 4. Sporicide. Proper use of sporicides can result in a 100% killing of all bacterial spores. Following are some common sanitizers of choice used in GMP facilities. 70% V versus alcohol, for surfaces, quaternary ammonium compounds, quats, phenolic sporicides. For killing spores, chlorines, such as hypochlorite, hydrogen peroxide, peracetic acids. Sodium hydroxide, cleaning and partial sanitizing, vaporized hydrogen peroxide, VHP. You should be familiar with your choice of sanitizing agents and only use approved agents in the nominated strengths. In the realm of good manufacturing practices, GMP, there are clear and strict rules concerning the cleaning and sanitization of equipment and manufacturing facilities. These rules are designed to ensure that products are manufactured in a clean environment, free from residues or contamination that could compromise quality or safety. To uphold these standards, companies must adhere to specific procedures, validate their cleaning methods, and meticulously document their activities. First, GMP mandates that the cleaning process itself be thoroughly documented through written procedures. These procedures serve as the official instructions for cleaning operations and must be validated to confirm their effectiveness. Validation involves demonstrating that the cleaning methods reliably remove residues and contaminants under defined conditions. Once a procedure has been validated for a particular piece of equipment or area, it is then published and implemented as a standard practice. This ensures consistency and provides a benchmark for ongoing cleaning activities. Second, the validation of cleaning procedures is a critical component of GMP compliance. All cleaning methods should be validated under the specific conditions in which they will be used. These conditions are detailed within the written procedures and include factors such as the concentration of cleaning agents, water type, pH levels, temperature, and contact time. It is imperative that employees strictly follow these validated conditions. Any modifications, such as adding more sanitizer or altering temperature, can invalidate the cleaning process. For example, increasing the amount of sanitizer beyond the prescribed concentration might seem beneficial but could actually alter the pH or chemical properties, rendering the cleaning less effective. Therefore, only the validated conditions should be used to ensure the cleaning process remains effective and compliant. Third, the protocols specify the exact conditions under which cleaning and sanitation should occur to achieve optimal results. These conditions encompass various parameters, including the strength of cleaning or sanitizing agents, e.g., 2.0% V, the type of water or solvent used, the pH of the cleaning solution, water temperature, and whether equipment needs to be dismantled prior to cleaning. Additionally, the procedures specify the required contact or residence time for sanitizing agents on surfaces, the agents used for removing residues or sanitizers, and the quality of water used in the final rinse, often requiring purified water for injection in GMP settings. Ensuring these conditions are precisely followed is essential to achieve effective cleaning and prevent contamination. Finally, maintaining detailed cleaning and sanitation records is a fundamental GMP requirement. These records serve as objective proof that cleaning activities have been carried out properly. They should document specifics such as who performed the cleaning, when it was done, and the methods used. For automated cleaning processes like Clean in Place, CIP, the system typically monitors and records cycle conditions automatically, including temperature, flow rate, time, and chemical concentrations. These records often include alarms and logs that indicate if any parameters deviate from the validated settings. Once the cleaning is completed, the records must be signed and attached, providing tangible evidence that the cleaning process was conducted according to GMP standards. This comprehensive documentation ensures traceability and accountability, reinforcing the integrity of the manufacturing process.